welcome everyone to the One World, One Health Update Rickettsioses webinar. This is the 14th live webinar session. Thank you for joining us live on social media. Just some introductions and some housekeeping rules. For question and answer, like usual, please type your questions in the Slido app. We will try to address as many questions as possible. With regard to CPD points, all frontliners, healthcare professionals, and allied health team, remember to collect your CPD points by filling up the online attendance form. This website link was broadcasted on the screen before we started the session. In case you missed it, the link is on our Twitter, Facebook, and YouTube. Please double check your email address before submitting. For the slides, notes after the session, after the webinar, the presenter slides, a summary of the panel discussion, and the Q&A will be made available on all our social media, websites, and email newsletters. If you'd like to rewatch this session, you can go to our clinical updates in COVID-19 YouTube channel, listen on our podcast channel, or you can read the ebook when it's available. With regards to today's webinar, we want to discuss about One World, One Health Update, which is Rickettsioses. And for the panelists for this week, I'm Dr. Suresh, I'm the moderator, my ID physician, Kinabalu. And we have Dr. Timothy William and Dr. Mohammad Yazid Abdad. Just some brief introduction for both of them. Timothy William is a fellow colleague, he's a senior clinical researcher, he's with the Malaysian Ministry of Health, CRC, Kota Kinabalu, from 2008. He's also head of Infectious in Gleneagles, Kota Kinabalu. He's the president of Infectious Society of Kota Kinabalu, Sabah. He's a fellow of Royal College of Physicians, Edinburgh, and honorary research associate with the Menzies School of Health Research, Darwin. He was awarded the 2017 Merdeka Award for his work on Knowlesi malaria. His research interests include malaria, tuberculosis, Rickettsioses, milioidosis, and dengue. With regards to Mohammad Yazid, our second speaker, he's a senior scientific officer in the Infectious Research Laboratory in the NCID in Singapore. He's a senior scientist there and provides scientific biosecurity, biosafety advice on ongoing projects. He's also actively involved in a number of projects as that's involved SARS coronavirus, COVID-19, CREs, bacteriophages, respiratory diseases, and of course, rickettsial diseases. Since his research career has begun, Yazid has published over 27 papers in peer-reviewed journals and authored a book chapter in Rickettsia. He has discovered a new species of bacteria, Rickettsia gravicei, and tick Ixodes wiley. He's also isolated and characterized the first description of Burma forest virus outside of Australia. And for today, we'll have these two speakers talking about this. And the first speaker, Timothy William from Gleneagles, Kota Kinabalu, will talk about Rickettsiosis as major etiologies of unrecognized acute febrile illness in Sabah, East Malaysia. With that, I will pass it on to Timothy to start his presentation. Timothy?
Thank you, Dr. Suresh, for the very kind introduction. I hope you all can see my slides now. Okay, I'll be presenting on our prospective study that we conducted in Kota and Kinabalu in Sabah on Rickettsioses as a major etiologies of unrecognized acute febrile illness in Sabah. And these are our list of investigators and authors for this paper. It was not an easy project to do, and I'd like to convey my appreciation to everyone in the team. So this is a bit of background on acute febrile illness in Southeast Asia. We have success in reduction in human-only malaria species. However, the etiology of other causes of acute febrile illness is still very poorly understood. There's difficulties in diagnosis, there's unclear epidemiology and treatment protocols. And acute febrile illness review in Southeast Asia, there have been 100 studies since 2012, whereby children, dengue accounted for about 50%, leptospirosis 27%, Orange Susugamushi 23%, and Salmonella typhi 20%. Children alone are dengue, Staph aureus, then non-typhoidal Salmonella, Strep pneumo, Salmonella typhi, Orantia, Susugamushi. So this is the paper in the Lancet of Global Health on 927 clinical presentations among 815 adults. I just want to go through here. For acute systemic infections, no pathogen was identified in 127 patients. So a substantial proportion of patients with acute systemic infections, as you all know, even in your own practice, we are unable to identify the pathogen. And in this case, there were 7 cases of scrub typhus, Orantia, Susugamushi, and SFG rickettsia. I apologize for this, because these are all tongue twisters. So just a few cases that were detected, and among the acute respiratory infections, there are only about 14. Now, what about Malaysia? Serosurveys have documented Rickettsiosis in Malaysia. Scrub typhus, the most frequent in urban areas. SFG are in rural areas, and the typhus group, Rickettsiosis, are unknown. Rickettsiosis are nationally reported, but no routine diagnostics are available. I think we all share the problem of getting a proper diagnosis of rickettsia. There were only 53 infections that were reported nationally, 2 of them in Sabah during 2009 to 2015. So, you know, only 53 infections for this notifiable disease. And Orantia Susugamushi or scrub typhus, nationally 0.6 cases per million people nationally, and 0.8 in Sabah. These were the ones that were reported. So this was the last big study that actually was done in Malaysia on febrile illness. This was done in 1984 and presented in a paper in the American Society of Tropical Medicine and Hygiene. And in this paper, this was more than what, 20, almost 30 years ago, where scrub typhus was the most frequent diagnosis, 19.3% of all illnesses, followed by typhoid and paratyphoid, leptospirosis, and malaria. So you can see even back then, scrub typhus was most frequent diagnosis. However, now we see that we seldom make a diagnosis of scrub typhus. So the transmission of rickettsial infection is by a mite, Leptosporidium, and it's all carried by rats and rodents. These are our study methods. We enroll patients from 2 district hospitals in northern Sabah, East Malaysia. Kudat, the population was 86,000, and Kota Marudu, 69,000. Febrile patients of more than 38 degrees, patients with non-malaria AFI from December 2013 to January 2015. So approximately just slightly more than 1 year, and reference standard testing for rickettsiosis. These are the study sites for Kota Marudu and Kudat in Sabah. These are the district hospitals. So our study methods, all patients were referred to the study if they were having fever of more than 38 degrees. We included patients who are febrile within 48 hours of admission, excluded those who are afebrile, and of course those who refused lost. Those who refused are lost to follow up. So in the first visit, we did an examination, clinical history, epidemiology, lab investigations, serum whole blood and whole blood, and all Buffy blood samples. And we asked them to come for a convalescent visit, a clinical history, examination, and serum blood sample collection was done. These are our diagnostic methods. The sera was screened by immunofluorescence assay for IgG 1 in 80, OT, SFGR, and TGR. So just remember the short forms for it. Convalescent and if positive, paired acute sera. Positives were titrated to 2,560. Acute phase sera screen at 1 in 40 for IgM by IFA. And we also did PCR for OT, SFGR, and TGR. So as you can see, 557 patients were screened. 426 patients were enrolled, both in Kudat and Kota Marudu. 131 were excluded. Those who attended follow-up were 183. We had available samples for acute phase, convalescent phase, or both serum samples, and 128 were whole blood or Buffy code. However, 243 did not attend follow-up. So for them, we had 240 acute phase samples and 221 whole blood or Buffy code. And then we had acute and convalescent phase or paired serum IgG serologic testing, convalescent phase IgG serologic testing only, PCR, acute phase IgM serologic testing, and acute phase IgG serologic testing only. So these are the results. Testing for IgG by IFA confirmed rickettsial infection. So these were the total number of those who were acute and past infections. 126 out of 354, 36% of patients were positive. Scrub typhus, 96 or 27% out of this. Spotted fever group, 26 or 7%. Typhus groups, Rickettsiosis, 25 or 7%. And we had patients with 2 infections, OT and SFGR 6, OT and TGR 8 patients, SFGR and TGR 1 patient, OT, SFGR and TGR 3 patients. So 36% of these patients who enrolled had either acute or previous infection, rickettsial infections. So these are the confirmed acute ricketsiosis, defined as paired 4-fold IgG titer rise to more than 160 in 38 out of 145, which are 26% of the patients. Okay, so they had acute confirmed, these were confirmed cases, confirmed acute ricketsiosis, OT 23, SFGR 9, TGR 4, OT and SFGR 1, OT and TGR 1, and PCR results were positive in 11 out of 319 patients, or 3% of the patients. We did PCR, OT 1, spotted fever 8, and typhus group rickettsia 2. Possible, unlike the confirmed cases, for possible acute ricketsiosis, we had 37 out of 354, or 10% of the patients. So these are the distributions. This is the tip Borneo, Kudat and Kota Marudu. These are the regions that where we had the cases. So they're all distributed around the surrounding areas of that supply the district hospital. So these are the demographics confirmed and probable. The males accounted for about 63 patients of confirmed acute rickettsial infections and 62% of probable acute rickettsial infections. Children, 16 in the confirmed group, and 8 in the confirmed group and 3 in the probable group. This was statistically significant p-value for confirmed or probable acute versus no infection. So what are the signs and symptoms of these patients? Headache and dizziness. The p-value for confirmed or probable acute versus no infection was significant. So many of them presented with headache, dizziness, hearing loss, and also vomiting. But of course, you can see this whole list of signs and symptoms were actually represented in the infections, which means to say that we cannot differentiate this disease from dengue or leptospirosis or malaria or others just based on the clinical signs and symptoms alone. One notable, we did not find any cases of escarp, which is classically described in Rickettsia or scrub typhus. Okay. The rest of the signs were actually very non-specific, which you can also find for other infections as well. These are the epidemiological features: Rickettsia infection, acute or past versus no Rickettsia infection. Look at the age was statistically significant. Most of them were 43 versus no Rickettsia infection at 34. Children less than 15, 9% versus 32%. Resides in a village, 67% no Rickettsia infection, 84%. And they were rubber tappers, farmers, and spend more time in the forest. So these are some of the risk factors of a person getting Rickettsia infection as compared to people who did not have Rickettsia infection. Okay. Laboratory features comparing Rickettsia with non-Rickettsia. Again, we really couldn't find much difference. Of course, we look at neutropenia. It was not statistically significant. Less than 1.5, we only had 1 out of 44 cases that showed neutropenia. Thrombocytopenia, 11 or 23% of patients versus non-Rickettsia infection. You get to see thrombocytopenia, but you cannot really differentiate it from the other infections. And acute kidney injury. Okay. No patient had a provision. None of these patients had a provisional diagnosis of acute Rickettsia infection. So no one was diagnosed as acute Rickettsia infection, nor were they suspected of having so. Now, diagnosis among those with confirmed acute Rickettsioses includes dengue, 14%, 12% were diagnosed as acute undifferentiated fever, 12% community acquired pneumonia, 10% urosepsis, 8% leptospirosis, 8% gastroenteritis. Among the Rickettsial cases, only 2 or 4% actually received doxycycline. Okay, so they were not diagnosed as Rickettsia, but they received doxycycline, but they received it because they had a provisional diagnosis of gastroenteritis. So the number of cases are likely underestimated. Mild cases may not have been admitted. They were treated in the outpatient and probably discharged. And severe cases, we also do not know. They may have been transferred to Queen Elizabeth Hospital or may have died. So this is something that a research gap that we need to look at the patients who are ill, who may have Rickettsia. Now, the mortality for cases that are untreated can be as high as 7%, not based on this study, but based on other studies. If we don't treat Rickettsia, about 7% of people may actually die. So these are conclusions. Now, this is important. The adjusted incidence Rickettsiosis is 15 cases per 100,000 people per year, compared to reported incidence of 0.08, which means to say it is more than 200 times, roughly about 200 times more than what is actually reported. Rickettsiosis are a common cause of AFI, 26%. Okay, 26% is quite a substantial number, particularly scrub typhus. The clinical and epidemiological features are non-specific. Acute phase IgM is insensitive for acute infections. So we need accurate point of care tests, and unsuspected Rickettsiosis are untreated, and this is dangerous. So therefore, we need improved diagnostic tools, and we need it urgently. In their absence, clinicians need to have an increased clinical suspicion and consider using empirical doxycycline, especially for people who have high risk of developing the disease. And this might avert disease progression and death. We also need to do prospective studies with PET serology to determine the burden of untreated Rickettsiosis across Malaysia. And also the earlier paper that I showed you in 1983. We have made more cases of Rickettsiosis than we know. So with that, I'd like to thank you very much. And I would like to acknowledge and thank our patients who participated in the study. The funding came from our Ministry of Health, the Australian government, and we'd like to thank the organizations as well, CRC, Menzies, and IDSKK, and these all our collaborators, Dr. Giri, Dr. Goh Pik Pin, Sinyo, Nick and Steve, and of course Matthew Greg, Steve Damner, and Megan Rella. With that, I'd like to thank you very much for your kind attention.
Thank you, Timothy. Leptospirosis, many of us have not been diagnosing it. In fact, since we are not suspecting it, we are not sending for blood investigations, and increasingly the labs are actually discontinuing the tests that is required to diagnose rickettsiosis. I think your study will help us bring back rickettsiosis in the map so that our diagnostic capacity comes back again. And I think the other thing that we will have to look at will be looking at the severe cases and see how much of mortality that rickettsiosis is causing, and we are missing. What I will do is I will move on to the next speaker, and then we can handle questions together from the public. So the next speaker, of course, is Mohammad Yazid Abdad from NCID Singapore, and he is going to talk about rickettsiosis, out of sight and out of mind. Yazid, please.
Thank you for inviting me, and thank you, Suresh, for the introduction. Good afternoon, everyone. My name is Yazid Abdad, and I work with the National Center for Infectious Diseases in Singapore. Asked to do a talk on rickettsia and the epidemiology of it. So I've come up with a quite simple, straightforward, hopefully something that everyone from a general, just to quickly introduce rickettsia. Now, rickettsia organisms are not new to science or medicine. We've known about it since the very start of the 20th century, and we have attributed it to a lot of outbreaks, large epidemics in history, from the Black Plague to typhus outbreaks in 2 years. I've seen courses where rickettsial information is actually disseminated to medical students. So it's not something new, and it is part of the coursework in many of the medical schools, renowned medical schools in our region. In 1910 as a result of Rickettsia typhi, which he was working on and trying to isolate. He did successfully isolated it, and because of his work in discovering rickettsia and sacrificing his life in his dedication towards discovering and uncovering a lot about rickettsia, the genus itself was named after him.
I think Dr. Yazid just disconnected.
So we will wait for Yazid to come back online. While waiting for him, we will use the opportunity to drill Timothy regarding this. Timothy, I was surprised that many of them did not get treatment with doxycycline. Is it because of azithromycin? Because we use azithromycin for many of the infections. Do you think that's why without doxy there was no adverse outcome in your study group?
No, I don't really think so, Suresh, because azithromycin is more difficult to get in the district hospitals.
Okay.
So it's not because of that. I think they were probably treated with ceftriaxone and all that, and they got better on their own, Having said that, we do not know the number of cases that were transferred to tertiary hospital, Queen Elizabeth Hospital, patients who didn't get better. So that's why it's important to suspect, I know it's difficult to diagnose, but to suspect rickettsia, because there's a therapeutic difference in the way we treat. We will give a different antibiotic. So my concern is, how it is, if ceftriaxone doesn't work, then we go to tazocine and we go to meropenem, especially in the tertiary hospitals, when the patient doesn't recover. So, and when that happens, it could either be a viral infection, but we want to show that in the district hospital, they were ill enough to be admitted, so if they, you don't simply admit patients. So these patients are ill enough to be admitted. And we want people to, we hope that people will consider this even by the roll of a dice, by the roll of a dice, if it's non-malaria and non-dengue, and we can't get a microbiological diagnosis, there's a high chance that it may be rickettsia. So consider using doxycycline, and actually in very severe cases of rickettsiosis, we actually need IV doxycycline. Since we don't have IV doxycycline, to consider IV azithromycin if all your cultures come back as negative. All your cultures come back as negative.
Just to the audience out there, please type your questions in the Slido app, and we can address your questions as they come in while we're waiting for Yazid to get back online. So when I treat rickettsia, what I used to think of is normal white cell count fevers. If they have, if they're not dengue, if it's not malaria, rickettsia is something that I used to think of. But I see in your study the white cell count didn't come out very significant between the two, isn't it?
Yes, that's one of the things that were, the classical view is that in these diseases, the white cell count will be low. The white cell count will be low. Having said that, when we look at it, the white cell count was not low. It was normal, but it was not high either, Suresh. Okay. You didn't get a leukocytosis compared to let's say you get leptospirosis, where often you have a leukocytosis.
Okay. So, okay, I think Yazid is back online. I think we will let him start again. Yazid,
okay? I'm sorry about that.
Okay, no problem.
Yeah. So which slide did I stop at? Pardon the cutoff. Yeah.
I think that you showed us Mr. Rickettsia.
Okay.
Ricketts.
Oh, okay. So slide 4, right? Yeah. Okay. If you can go to slide 4.
Okay. The next slide. You can start the next slide.
Yeah. The next slide. Okay. The one before, I think. Before.
This slide.
Before number 4. That is slide 6. No, this is
okay. I think
okay, before this one. Okay, was it this one? Okay, we can start from here then. Okay, so sorry I got cut off. So what I was saying is rickettsial organisms are part of the alpha proteobacteria group of bacteria. If you are familiar with the taxonomy on how bacteria are classified. So alpha and gamma proteobacteria are generally, for the most part of it, intracellular bacteria. So some of the genera listed there would be very familiar to you, such as Rickettsia and Aurantia, which Dr. Timothy has just talked about earlier. Some of you may be familiar with Wolbachia, which is being used in Singapore and Australia to neuter mosquitoes or to control mosquito populations. You have Ehrlichia and Anaplasma, both are zoonotic organisms, mainly from dogs, and also Coxiella burnetii, which is a huge problem for the cattle industry, okay, and a lot of other livestock as well. So a lot of these organisms are intracellular bacteria, and what that means is that it requires a host cell to reproduce. It's unable to survive or last long in the environment on their own, which is not unlike viruses. However, the major difference between viruses and rickettsias is that Rickettsiosis have got a proper cell wall and a cell membrane, and they have got nucleus and also vacuoles. However, they are not able to reproduce on their own. They have to hijack their host's cellular machinery. Next slide, please. So work on Rickettsiosis are restricted in BSL-3 laboratories worldwide. Okay, this is quite a standard. And the main reason for this is that at some point in history, a number of countries has actually tried to weaponize Rickettsiosis. And one of the main Rickettsiosis that they attempted to weaponize was Rickettsia prowazekii, along with others such as Coxiella burnetii and so on. And thus, countries like Singapore, the US, they require Rickettsia to be worked on in a secure and protected location. Now, Rickettsiosis, though we don't really hear much of it in the general media and so on, is actually widely distributed worldwide. It's found on all continents except Antarctica, and mainly acarids and insects. And acarids refers to ticks and mites. Infection can occur from direct contact with infected materials, such as the fecal matter that fleas leave on your skin. So Rickettsia typhi is actually transmitted not from the flea bite, but actually from the fecal material on your skin where it enters the pores, or you have a cut on the surface of your skin. And thus it requires exposure to the open epidermal layer or mucosal surfaces. And thus laboratory-acquired infections are typically quite rare. However, it has occurred in the past. Next slide, please. So this map is from a publication from me and colleagues recently where we reviewed the work done by our other colleagues worldwide. And this sort of tries to summarize the different Rickettsiosis worldwide. So if you look just alone within Asia itself, there's a number of Rickettsiosis that do afflict us potentially. Here in Southeast Asia, we are looking at least 5 to 6 different rickettsial species. Some of them is Rickettsia helongjingensis that was originally discovered in China. Rickettsia japonica, as the name implies, it's from Japan. And we also have Rickettsia conori, subspecies indica, conori, and israelensis, and also Rickettsia honii. Orange-shirt Susugamushi is a big problem here in Southeast Asia. As I mentioned in the slide before, during World War II, it was one of the major causes of mortality and morbidity among soldiers, aside from malaria and dengue. Okay, next slide, please. Okay, so restriction on containment level makes Rickettsia challenging for scientists and clinicians to work in. And the disease cause generally called spotted fever, typhus fever, or scrub typhus. And it depends on the organism that infects the patient. However, diagnostics of rickettsial disease can be a challenge if you are trying to differentiate between the 3 different pathogens, types of pathogens. So the next point, transmission via bites, I've already mentioned before, by hematophagus vectors. Sorry if I'm using big words here. But hematophagus vectors mainly mean they are insects that bite and consume blood from their host. And when I say host, it doesn't have to be humans. You can also refer to animals. One thing to note is that the disease does not require a high infectious dose. It can just be caused, only requires a single organism to get past the immune system to eventually cause disease, which is known as Q fever. Okay, next slide, please. Now, the reservoir for rickettsial pathogens can either be the vectors that transmit them themselves or small mammals that inhabit, that place host to the vectors. Work has been done to investigate the role of dogs, wild pigs, and various other feral animals, large mammals, if they can be viable reservoirs. But in most studies, it has been shown that such large animals are not sustainable. Vectors tend to be not specific for each rickettsial species. However, there are some rickettsial species that has been reported to be transmitted by only a single vector species. Now, we believe the reason for this is because they have shared a significant period of time evolving together. And at some point, they have sort of evolved and have a form of symbiotic relationship. And thus, they are not able to transfer or be carried by other vector species. Now, reservoir transmission, which is transmission from one tick to another via shared feeding area, sexual transmission, and so on, and also vertical transmission from parent to young. Next slide, please. So this is to show a picture of a feral pig. So that's the face of a feral pig that we were doing a study on back in Western Australia many years ago. And what I'm trying to highlight here is that there's two different tick species close to each other and they're sharing a single bite site. Emblyoma trigotatum and Etiologies australiensis. Both of them are known to infest the large mammals, especially the kangaroos, the woylies, the quokkas. And Emblyoma trigotatum has got 100% prevalence for Rickettsia grazii, while Etiologies australiensis has probably got a prevalence of anywhere between 5 to 30%. When I took back both ticks to the lab, both of them were positive for Rickettsia grazii. So as you can see, just sharing the bite site would allow for the transmission of the bacteria from one tick to another, and thus establishing that other tick potentially as a vector for the disease. Yeah. Next slide, please. Thank you. Now, Rickettsioses, as Dr. Timothy William had said, shares many symptoms with other febrile illness. Okay, one of the main markers is rash, and also, as he mentioned, eschar. However, he didn't see any eschars in any of his patients. So this is it is actually quite uncommon. It's actually quite common not to see any eschars in patients, and also not to be able to see any rash, unfortunately, as these would be very good markers to actually differentiate Rickettsiosis from other febrile causing pathogens. Now, thus, using syndromic diagnosis can be a challenge. As a result, we try or are trying to attempt various methods of diagnosis in the laboratory. However, a lot of these methods can be expensive and require a certain level of training, which is not available in most locations. Next slide, please. Thank you. Okay, so when it comes to the laboratory, there's many ways we can do rickettsial diagnosis. And depending on the depending on the patient sample that is available to you and what the patient is willing to give. So we can do diagnosis using dried blood spot. You don't have to require to do a bleed on the patient with a full blood tube. Blood serum biopsies, you can put them into culture to try and isolate. You can do PCR on various parts and types. Now, when we do diagnosis of patients, usually in a lot of countries that are aware of rickettsiosis, in the US and Australia, they would encourage the patient to bring in the tick that they pulled off or caused the eschar to appear. Just like similarly, if you are bitten by a snake, they expect you to be able to describe the snake, or at the very least, if you can't describe the snake, try to identify or see what the snake looks like, and so on. Right? So there are various methods to do it. However, a lot of this require quite significant investment by the laboratory that wants to introduce rickettsial diagnostics. And if the laboratory, however, has already got this in place, then to be able to do it will be quite relatively easy. Okay, next slide, please. So one of the main ones, the gold standard, is IFA, immunofluorescence assay, as Dr. Timothy Williams has used for his study. This is part of diagnosis using serology and recently the use of ELISA has becoming more widely accepted and more popular among some of the laboratories I collaborate with. So to the right, you can see a picture of an IFA, and the trick to this is that a lot of new staff that we try to train usually are unable to differentiate between background staining and the actual rickettsial bodies. If you see the little bright dots, that are actual rickettsial bodies themselves. They're really tiny and are approximately 2 microns long and about 1 micron wide. Okay. Now with IFA, to have a diagnosis that the patient was infected by a rickettsial pathogen, they need to have a convalescent sample with a 4-fold rise in antibody titer. However, with IFA, we are unable to determine the species that caused the disease because there's a high cross-reactivity rate between species of the same serogroups. So the serogroups, just to remind you, are typhus group, spotted fever group, and scrub typhus. Okay. So the other method is by PCR, polymerase chain reaction, and there's a lot of real-time PCR methods that you can get from published methods. However, I've not seen any kits that actually are able to differentiate Rickettsiosis. And many of them, if they do, usually only do for SFG, NTG, and Orantia, but they are still also not able to differentiate between species. PCR tend to be very, very sensitive, and you can detect presence of rickettsial rickettsial nucleic acids in mixes with 10 copies of the target or even less in some assays. If we need to differentiate the species, then we can do downstream sequencing. Yeah, thank you. Next one, yeah. Cell culture is something that we are trying to encourage. Not many laboratories do cell culture because, as I've highlighted before, of the security restrictions with rickettsial work. And not many people do cell culture nowadays. It requires a high level of competency, unfortunately. And the other challenge is the very long doubling time. A single rickettsia cell would reproduce and become 2 over an 8-hour period. For just to compare with E. coli, Escherichia coli, within the same period of 8 hours, a single cell will become 16.8 million. Okay, so trying to isolate rickettsia can take months. Just to be able to see if your culture is successful and you've got cytopathic effect, which is what happens when your whole cell starts to die. All right, next slide, please. So this is a bit blurry, sorry about that, but I was trying to show you what cytopathic effect means. So this is a picture from the ATCC, American Tissue Type Collection. And this is a picture of Rickettsia assembiosis. It's grown in C636 cells, which is Aedes albopictus. So on the left, you have a confluent monolayer, and on the right is what happens when Rickettsia assembiosis has infected the cell line, and you have a lot of cell death happening. So a lot of the empty areas that you're seeing is where the cells have died off and lifted off from the confluent monolayer. Okay, next slide, please. Thank you. Now we move on to treatment. Before the discovery of antibiotics in the US, the mortality attributed to rickettsial diseases was as high as 70%, and in some locations even higher. Rickettsia rickettsii, and I think that its close cousin Rickettsia parkeri, which was discovered only recently in the 80s, highly virulent and cause high mortality compared to other ricketsias in our region, such as Japonica and Hornii and so on. The treatment is still the same. As Dr. Timothy Williams have mentioned before, you can treat with either doxycycline or chloramphenicol, and it's usually very highly effective. However, that's the challenge, because once you treat the patient, even after just the first dose, half an hour after the first dose, the sample that you collect from the patient is useless. You can't use it for diagnostics, especially PCR, because the antibiotic is extremely efficient in getting rid of the organism. For more serious cases, it is recommended that patients are given doxycycline intravenously. Now, studies recently have shown that there's no signs of antimicrobial resistance. I think this can be attributed mainly to the biology of the organism itself. Because of the long doubling time it requires for it to reproduce and multiply, it's not able to, any changes, significant changes to its genetic code is not established well enough. So we go to, I believe, the third last slide, which is the challenges faced with rickettsial work. So as highlighted before, the challenge is in diagnosis. Samples have to be collected within very narrow windows. Okay, so example, if we use IFA serology, you can only diagnose and confirm the patient has had rickettsial infection 2 to 3 weeks after. So you actually have to start doxycycline on suspicion rather than treating them as a response confirmed after diagnosis. PCR requires a very narrow window where the patient has to have the sample collected within the first 1 to 5 days of symptoms showing. So thus we are looking at developing a more robust and efficient diagnostic system. When I say we, I'm referring to the Rickettsial Pathogens Track Reduction Network funded by DETRA, which is a conglomerate of rickettsial scientists from around the world. And it includes a lot of experts from the US, Australia, Singapore, Malaysia. I've got some colleagues in Malaysia involved in that as well. And we are looking at developing quite a number of assays at the moment. Another challenge is that in our region, a lot of information is unknown. We need to encourage or try to allow for rickettsial disease to be included in studies where possible to better understand it alongside other diseases that cause febrile illness, similar to what Dr. Timothy Williams have done. A lot of studies in our region has not been targeting rickettsioses by itself, but that's understandable because the number of cases caused by rickettsial disease is a lot lower than other more pressing diseases such as malaria, dengue in our tropics. However, the challenge again is then we don't know what's causing disease out in the bush, in the rural areas. Just to highlight example, Rickettsia honii has been known to be in Thailand for many years. However, its prevalence in Thailand is unknown. The bacteria has never been isolated in culture. So a lot of the information that is known about Rickettsia honii in Thailand is as a result of small studies that uncover or discover information about Rickettsia honii by chance, serendipitously. Now, in many countries, in some countries that I know, rickettsial disease is a reportable disease, which I believe it is in Malaysia. It is in Singapore. However, some of our neighbors, how much of their population is actually being afflicted, and if cases coming to the hospital is recorded, whether they're rickettsial disease or not. Now, in the effort to expand on work on rickettsioses, biosafety is still a major concern, and laboratories need to do good risk assessment to ensure proper measures are in place. So trying to put together a BSL-3 laboratory can be cost prohibitive. The maintenance, the equipment, the training, and so on. Okay, next slide, please. So hopefully what I presented have given you a brief introduction into rickettsias. Fortunately, we have 3 reference laboratories worldwide. They are very helpful. They are very willing to support and how do you say it? Encourage research. There's one in Australia, Geelong, Victoria. That's led by Dr. John Stenos. There's the French lab led by Professor Pierre-Edouard Fournier. And the last one is the CDC itself in Atlanta, Georgia. I've worked with Professor Pierre-Edouard Fournier and Dr. John Stenos very closely in the past. I've visited their labs. I actually spent 3 years in Australia with Dr. John Stenos, and I think about a few weeks with Pierre-Edouard in his lab. And they have a lot of resources that they are very willing to share with colleagues around the world to try and help them understand their rickettsial problem locally. Okay, and next please, next slide. And that's my contact. You are more than welcome to drop me an email if you have any more questions outside of today. And if you need further information, please, you're more than welcome to reach out. Thank you.
Thank you, Yazid. I think we'll move on to, we've got about 15 minutes to go through some of the questions that has come out in Slido. I think the first question is asked, what specimens were used for the PCR for Rickettsiosis? Is serology the gold standard for diagnosis? Anybody wants to take it on?
Yeah, sure, I can do it. So for serology, it's serum or plasma. So if you're working in the hospital, you can either use the EDTA blood tubes, will give you plasma, or the gold top tubes, which the SST blood tubes, will give you serum. Or if you don't have the gold colored liquid from the top, and that can be used for serology. For PCR, if it's from a patient, you can either do a PCR of the Buffy code. So if you're not familiar with the Buffy code, but you know what blood tubes are, if you look at the EDTA blood tube, the purple top ones, if you do a centrifuge of the tube, you have a layer of white color material between the plasma and the clot. That's the Buffy layer, and you can do a PCR on that after DNA extraction. The other option for patient would be to actually biopsy the eschar. So if you have an eschar, like I showed you in the picture of the patient in Western Australia, if you can get permission to do a biopsy, you can do a PCR on that, and you should be able to get a high amount of Rickettsiosis. Now, a colleague of mine has actually shown that you don't have to do a biopsy because that can be very painful for the patient. So swabbing the eschar and doing a PCR of the actual swab of the eschar itself should be able to help you with a diagnosis of it by PCR. Yeah.
As a follow-up question, Yazid, in clinical labs, the PCRs are not widely available. Increasingly, the reference labs, at least in Malaysia, are discontinuing. We used to have IIP, indirect immunoperoxidase test, which seems to be being discontinued. Is ELISA, commercial ELISA available, and is it useful?
Okay, commercial ELISA is not available. There are, however, kits on the web, such as the ones by Penn. I think Penn Bio stopped making their kits. There's just no business for it. There are other kits that a colleague of mine, George Vargas, I think he's at the Catholic College in India, has recently looked at, but I can't remember for the life of me the name of that kit. But I can share it with you guys later once I picked it up. There are some kits that is not as good. There are some that are well designed, but how long they'll stay on the market, I can't guarantee, and I can't put any recommendations out. Now, the list of the laboratories that I showed you earlier, a lot of, like Singapore, we send our samples to the lab in Australia. The cost is quite low. It's quite affordable. And if you need to develop or implement your own assays locally, I should be able to assist with that as well and point you to the right papers to use or the assays to use and develop it locally. It will be a lot cheaper than buying kits.
Okay, thank you. Yeah. Next question to Timothy. As a district hemo, how do we decide when to prescribe doxycycline?
I think, okay, so a patient comes in with fever, undifferentiated, so you don't really know which particular organ is involved. So you just coming with fever alone. So we normally have a few differentials: dengue, malaria, typhoid, scrub typhus, leptospirosis, isn't it? That's our usual, compared to, you know, a person coming with frank pneumonia or urinary tract infection or a boil or a skin infection. So when a patient just comes in with fever alone with no organ involvement or multi-organ involvement, and they are negative for dengue and they're negative for malaria, then I would consider using doxycycline. Maybe if they're ill, then I'll give doxycycline and ceftriaxone for these patients. So that would broadly cover most bacterial infections. Of course, don't forget about melioidosis as well. So there's no real hard answer that I can give. What I can say is if the patient not responding to the usual antibiotics that you can give doxy, or you can give doxycycline if there are no contraindications as the first line drug for these patients who come in with undifferentiated fever.
I agree. I mean, the other thing, of course, is I think your study also showed, rural background, epidemiological history.
Yes, exactly. True. So if you're coming from a district hospital, as compared, you're coming from a city, like Kuala Lumpur or if you're coming from a place where there's rubber estates or palm oil or fringe of forests, which people were working as farmers and all that, that epidemiology history will actually assist you a lot.
Any reason, next question, of course, both of you can try and answer this. Any reason no ESCA was found?
I'll answer first. I think, first of all, you have to look hard for an ESCA, isn't it? You actually have to look for it, the folds of the skin and all that. And it may be small, it may be big, or people may just miss it. So that may be the reason why an ESCA is not found, because you didn't even suspect rickettsia, isn't it? So when I'm not using, and you really need to actually expose the patient, take out all the clothes and look at the folds, the groin areas and all that. So that may be the reason why, that might be the reason why ESCA is not found. But in any case, there may not have been an ESCA. As it said, ESCA is most likely not there in the first place.
Yazid, anything?
Just to add, I can add a bit. This is something that me and my colleagues in the field of rickettsia has discussed quite why an ESCA might not appear at the bite site. It could be because that the actual pathogen itself did not form a beachhead on the patient's skin, because an ESCA is basically an infection of the epidermal layer by the pathogen itself. So it may have directly entered the bloodstream and continued to infect the epidermal layers within or spread via the vascular system. So it didn't, aside from that, it could also be underlying causes. The patient may have a compromised immune system, so an ESCA may not develop. But and also the infectious dose might be also an attributing factor. But no evidence to support any of this. A lot of it is just conjecture because we are not allowed to experiment on humans.
And then the last question here in the Slido is any relation to post-tick bite Lyme disease in US?
No, because that's a totally different species. Lyme disease is caused by Borrelia. And as you know, ticks are notorious for carrying a lot of diseases. There are a lot of viruses that ticks can transmit. I think in Russia they've got a big problem with viruses that are tick-borne. And so Rickettsiosis versus Lyme disease, yes, they are totally different species, totally different bacteria. They may share similar vectors, they may share a similar mode of transmission, but the biology of both diseases are very, very different.
Thanks. Just a last question from me, Yazid. We clinicians sometimes, because we don't have IV doxycycline, sometimes you find us using IV ciprofloxacin or IV macrolized IV azithromycin and treat it, and we think it works. Any data for that?
Not that I'm aware of, but I think you should ask Dr. William, because I'm not a clinician, sorry.
Any susceptibility testing done in this particular group? in the lab,
Yeah, unfortunately, no one to my knowledge has done susceptibility testing for Rickettsiosis. Yeah. And also, yeah, there might be some studies looking at comparing the different antibiotics on how effective, but because doxycycline is so effective, people just fall back on it at the end of the day. Yeah, but there might be studies that I'm not aware of. Yeah, probably.
Timothy, anything else?
I think when we don't have doxycycline, IV doxycycline, the patient is very ill, out of desperation, and as you know, in the ICUs where meropenem doesn't work, then we don't know what to do. Then, if the patient fits the profile, the epidemiological characteristics and this and that, I think it's quite prudent, I think, to consider IV azithromycin. IV azithromycin when the usual antibiotics are not working.
Thank you for both of you, Timothy, William, and Yazid, for enlightening us on rickettsiosis. A nice change from COVID for a while to talk about rickettsiosis. I think with that I will end, and I will pass this to Dr. Goh, who wants to say something with regards to the whole NIH series that has been so wonderfully conducted so far. Dr. Goh.
Thank you, Suresh. Everyone out there, and Timothy, William, and Mohammad, and Yazid, we are finally coming to an end for this clinical updates of COVID-19. We have done 13 series of webinar on COVID-19, and this last one of Rickettsioses is very important. And I would like to thank audience who are participating, and certainly want to take this opportunity to thank a few people who are instrumental to this clinical updates webinar. First is Dr. Woon Yon Liang, who actually suggested this idea during the peak of COVID-19 pandemic. At the time, we see that many people want to know more about COVID-19, but we are just too busy managing COVID-19 in some hospital. With that, I actually got hold of Lakshman and then Suresh, and we got permission from our DG to have these bigger updates. So all this was done very peacefully, very fast, and this webinar will not be a success without the technical team consisting of Dr. Chu Cheng Hoon and her colleagues such as Yip Yang Yi, Tan Ming Sui, Lim Ming Sui, and a few others colleagues in the ICR, Institute for Clinical Research, and the ICT team in NIH. Without their hard work and this amazing talent in learning how to do webinar on this social media, and not only one platform, many platforms. I also want to thank CRC team in Sungai Buloh, because many of the speakers are under team, Dr. Suresh Kumar's team, and also CRC HKL. Besides that, the moderators, I'd like to thank Dr. Christopher Lee. He has been always very supportive and always behind us, and has been moderating most of the session. And also Dr. Hisham Shah, our Deputy Director General of Research and Technical Support, who had moderated a few sessions. This is incredible work. I think this teamwork is amazing. The IT technology has made us very close together. I sincerely hope that the team's webinar on COVID-19 provide you with updated information and very in-depth information from the clinician team, from the laboratories such as IMR and NKAK, as well as from the public health and the primary care, and we have now a foreign speaker. So from the bottom of my heart, I wish you all the best, and we all pray that pandemic COVID-19 will end in Malaysia soon. So Suresh can go back to his own way, own life. All of us can go back to living like before, but this is wish fulfilling. Hopefully is something that we'll realize soon, but if not, when the situation arises, when there are new topic, we can reactivate this group. So once again, I want to thank all this in front and behind the scene, and the team in NIH, in ICR, and in CRC, and the whole MOH team for this successful webinar series. Thank you again. Good afternoon.
So,
Yes, thank you. Bye. See you. Thanks, Timothy.
Thanks, Vipin.
Thanks, Izad.